Good afternoon, everyone. My name is Paul Burke. I'm the Chief Executive Officer at Northern Territory Farmers Association. This is episode 6 in our webinar series, Growing the Territory. Today we're going to be talking about fall armyworm, which has obviously made its way to the Territory and in very recent times. So there's a lot we're going to go through today. I'd like to welcome everyone online and acknowledge some of my previous colleagues from AgForce Queensland who I know are attending today. G'day. Just like to tell you that the Territory's open and we can go to the pub and stand up at the bar and have a beer. So I just thought I'd share that with you. Today I'm joined by Brian Thistleton, the Principal Entomologist at NT Department of Primary Industry and Resources. Dr. Praise, Senior Entomologist at Department of Primary Industries, and Michael Finney, Plant Biosecurity Officer. So without further ado, I'll kick off. Dr. Praise will go first. If there's any questions, please pop them into the comments box down the bottom and we'll try to get to them. If we don't get to all the questions, we'll answer them and put them up on our Facebook page with the recording of this. And in addition, there is a survey that we're trying to gather some information. So if you could fill that out as well, all of that information is collated. Can't say who it is, but it would be very helpful for us in discussions around fall armyworm. So without further ado, over to you.
Okay, so good afternoon, everyone. Today I'll be talking about the biology, distribution, host range, damage, and a bit of the characteristics of the fall armyworm.
Okay.
So I'd like to start with a brief background on the origin of this pest. So fall armyworm is a native to the tropical and subtropical regions of the Americas, and all year round it was observed that this moth lives as far as south as La Pampa, Argentina, to as far north as southern Florida and Texas, USA. So this moth does not survive under a prolonged freezing temperature. Therefore, there is a seasonal migration during summertime, and it is able to migrate as far north, as far as Quebec and Ontario. Because of its ability to travel over long distances, this pest has rapidly spread to other parts of the world. So in 2016, infestations were reported first in Nigeria, Ghana, Benin, and Togo, Africa. In 2018, fall armyworm was detected in India, and this has then spread to other Asian countries, including Myanmar, Sri Lanka, China, Bangladesh, Thailand, Korea, Indonesia, Japan, and the Philippines in 2019. And earlier this year, fall armyworm is detected in Australia. So here, this is a map showing the current distribution in Australia. So the fall armyworm was first detected in the Torres Strait Islands, and from the point of detection, it has been successively spreading throughout the northern Australia, and that includes the North Queensland, Northern Territory, and the Western Australia. So for Queensland, it has now reached as far south as Bundaberg, and for the Western Australia, it has also reached as far as Carnarvon. So in the Northern Territory, we detected the moths in the pheromone traps in Catherine, Tipperary, Douglas Daly, Coastal Plains, Adelaide River, East Point, and Berrima Farm. So moving on to the characteristics and behavior of this pest, fall armyworm occurs in 2 strains that are identical in its morphological features, but differs genetically based on larval host specificity. So we have the corn strain or C strain that feeds on large grasses such as corn, sorghum, and cotton. We also have the rice strain or R strain that feeds on small grasses such as rice, Bermuda, and pasture grasses. The pest is also considered a strong flyer where it can travel 100 kilometers per night, and no wonder it spreads fast all over the world. So fall armyworm is considered as well as polyphagous insects, and it was reported to feed on more than 350 hosts with preference for the Poaceae. So other hosts include plants from the family Fabaceae, Cucurbitaceae, Asteraceae, Amaranthinaceae, and others. So if you want to know the complete list of the 350 hosts, this can be found in Montesano paper in 2018. Another characteristic of this pest is that it has high fecundity. So a female can lay up to 1,000 eggs of its lifetime. Then this pest also survived better on warm and moist environment. Here I am showing you a map on the climate suitability for the fall armyworm, and it shows here that in Australia, the population of this pest is likely to persist in the northern Australia. As you can see here, the color yellow, orange to reddish. So this is where the areas where it is likely to persist. So all year round, this pest can live on these locations. And the green areas indicate that population may grow but not continuously, as population may die out during winter. Now let's talk about the life cycle of the fall armyworm. So the fall armyworm life cycle starts with egg, and they are laid in masses of approximately 150 to 200 eggs. So the eggs are pale yellow in color and laid on the underside or on top of the leaves, covered with protective layer of scales or setae from the female abdomen. And the maturity of an egg will take 2 to 3 days. The next stage would be the larval stage. So the larval stage occurs in 6 larval stages. So we have the first instar up to the 6th instar. So as you can see, the young larvae are greenish to brown with longitudinal stripes and black head. But sizes vary depending on how much food they have eaten. So the larvae of fall armyworm are similar to larvae of other related species. That's why in the field, it is very challenging, or it's hard to identify. But if you want to have a sure identification of this pest, molecular analysis is required. So as they develop, there is a variation in color, and the spots on the body is already evident. So in the 3rd instar, the dorsal surface of the body becomes brownish and lateral white lines begin to form. In the 4th to 6th instars, the distinctive characteristics of a fall armyworm are becoming clearer, with the head is reddish brown, mottled with white, and the brownish body bears in subdorsal and lateral lines. So to help identify this pest in the field, I have here a photo of mature larva with the description of its distinct characteristics. So a mature larva, a fully grown larva, is about 30 to 40 mils. So as you can see, there is an inverted Y on the head, and there are also light bands on the side of the body. And so you may observe as well that in the second to the last segment are 4 large spots arranged in a square shape, but there are also smaller spots in a trapezoid shape, which can be seen on the different segments of the body. So the body of the fall armyworm doesn't have dorsal or subdorsal patches, but there is a pale dorsal line that runs up and down their body. So the next stage is the pupal stage, and this is about 15 to 20 mils. So the color is reddish brown, and pupation usually takes place in the soil at 2 to 8 centimeter depth. So pupation is also observed on maize cobs. So If the soil is too hard, the larvae construct a loose cocoon using leaf debris and other material on the soil surface. And during the pupal stage, or the duration of the pupal stage would take 9 to 13 days. So from pupa, an adult, so moth emerged from pupa, sorry. And it has a wingspan of 32 to 40 mils. So adults are nocturnal and most active during warm, humid evening. So the duration of adult life ranges from 7 to 21 days. And the total development period or life cycle for fall armyworm is or ranges from 30 to 90 days. In terms of identification of adult moths, so male and female differ strongly in that females have less distinct wing markings, ranging from a uniform grayish-brown to a fine mottling of gray-brown. So the forewings of males are mottled, as you can see here, that they are also shaded gray and brown with orbital spots. So this is the orbital spot here. It is circled with red and with triangular white spots at the tip and near the center of the wing. So in the laboratory, we usually do dissection to have a positive identification of the fall armyworm. So this is how it looks like the genitalia of the fall armyworm. So we have the male genitalia and female genitalia. In the field, you may also encounter species that is related or closely related to fall armyworm, which is the false armyworm. This is Leukemia laurii. And it is the characteristic of this false armyworm is very distinct, as it has a very fluffy tail. So the color of the wings are brown as well, and it has a series of narrow longitudinal dark stripes with tiny white spot near the middle. So as you can see here, this is the genitalia of Leukemia laurii and the fall armyworm, and they look different. So if you want us to identify whenever you observe moths in the field, you can send us your samples and then we can do dissection. So in terms of damage, I'll be showing you here photos of damaged maize plants we captured in the Northern Territory. So basically, the larvae defoliate the leaves of the host plants, and the very common damage is skeletonized leaves of the plant, and frost can also be seen on the whole of the plant. So the larvae also make holes in the leaves, which is very visible, and caterpillars also burrow into the growing point or hole of the plant. So not only the fall armyworm damage the leaves, but they also damage the corn cobs. So this is a photo showing larval damage with frost on maize cobs. This is also showing you a photo of larval damage on cotton balls and larva feeding, larva damage on grasses. Okay, I guess that ends up my presentation today, and for inquiries, you can contact our office with this phone number and email address on the screen. So thank you very much.
Thanks, Dr. Praise. So obviously, with us trying to develop an emerging industry, fall armyworm couldn't have come at a worse time. So it's just really important that everyone considers surveillance as part of their normal business, stays in touch with Department of Primary Industries and Resources if they have got an infestation. And there are some well-developed fact sheets around treatments as well. So, you know, we're all trying our best to understand what the impacts will be, but there have been reports of some losses in crops. So it's something that we really need to watch and really need to be conscious of. I'm going to hand over now to Michael Finney, Plant Biosecurity Officer, to have a chat.
Thanks, Paul. Good afternoon, everyone. My name's Michael Finney, and I'm a Plant Biosecurity Officer, and I've been involved in the monitoring for fall armyworm since approximately November last year. So I'm just going to have a quick discussion about some of the practical components in terms of monitoring. So I just wanted to have a look at a couple of questions and just go through these briefly. So we'll have a look at what traps are required for monitoring for fall armyworm. We'll also have a brief look at what pheromone lures we use for the monitoring, and we'll look at the placement of the traps in the field. This is particularly important to get the best out of our monitoring, and we'll have a quick look at where we currently have traps across the territory, and then we'll just do a quick wrap up. So the first bit of monitoring is that we use these standardised bucket traps. And as you can see here by the two photos, number 2 just shows the whole trap, and number 1 just sort of shows the various components. So as you can see there, you've got a top half and a bottom half, and they just basically snap together. You can see just in the bottom of the image number 1, it just shows a little green cage. This is where we insert the pheromones into, and as you can see on the image there, they just sit in top of that hole in the trap number 1 there, and then that attracts the male moths into the bucket. We also put a small insecticide strip in the bottom of the white container, so as they fly in, they go in there and then die from there. So yeah, that just shows exactly those traps. So the best thing about them, they're really simple to use, and also in that the white container, we've modified the trap particularly for our wet season environment. So in that white component, there's a bit of wire mesh, and you can see there's a small hole drilled in the bottom. And that's because the moths can sit on that mesh, and the water can flow out the bottom without having the samples really wet and soggy, which are then aren't any good to identify. So these are the two main lures that we use. The image number 1 just shows the rubber scepter, and image 2 just shows the plastic card type. So the reason why we use the two different lures is because the companies will have a slightly different chemical makeup, but they are relatively similar in terms of their chemical structure, but just slightly different. So we're trying to use the two lures to determine if there's any kind of huge variance in trap numbers. At present, we haven't noticed a large discrepancy between the two, but that's something we'll continue to monitor as we go forward and do more trapping. A point that I want to touch on about the lures is it's really important not to contaminate the lures between the traps. So we always try and use gloves, disposable gloves, to not contaminate each lure and keep them nice and separate. So we're just going to have a quick chat about trap placement and site selection. As I mentioned earlier, this is really important to get the best out of your monitoring and really yield the best results out of these type of traps. So we're aiming for the male moth, and they're more likely to occur in areas where the females are present, and these will tend to be in areas where it's suitable host plants are. So that's a number 1 criteria, is to try and find areas where there's suitable host plant material. So photo number 1 just shows a trap location in Katherine. It's located near a cattle feedlot, and it's located between about a 40 hectare of irrigated sorghum, and it's also got another 40 hectares of irrigated Rhodes crops and assorted sorghums, which are soon to be harvested. So that's an ideal location to have one of those traps. And the photo number 2, again, just shows another image from Catherine. And this is located next to a wet season millet crop. And it's also got a crop which produces watermelons as well. And as you can see, it's been recently harvested for hay. So the image shows there, or both the images show, that the traps can be placed either in a tree, or they can be hung on a fence post or something similar. The ideal situation is to have it at approximately 1.5 metres just off the ground. And the traps should be at least 50 metres apart from each other. And that's because we're using those 2 pheromones just to increase the distance and stop any kind of cross-contamination from there. We also look at essential criteria like, is it safe to access the site? Because obviously we're going to be checking these things regularly, so we want to be able to access them quite easy. And like I said, also preferably on host plants or host plant sites, such as native sorghums, rice, and tall green grassy areas of some of our native species. Also, we want to place these traps in open areas, so it's not closed off by vegetation. So this allows the pheromone to drift out from these traps and encourage the male moths to come into those traps. Also, we want them to be in dark areas, so preferably away from street lights and other kind of light interference. And obviously a concern for us in the territory to avoid fire-prone areas, being the dry season at the moment. We know that regular burn-offs occur, so that's just something we need to keep in mind when placing these traps. So an important stage of monitoring is clearing the traps. So the photo number 1 just shows how in the field they're quite easy to clear. You just snap the 2 components. The white component's just held in by a couple of simple lugs, and you just undo those and see what's in there. So the photo number 2 just shows what kind of volume can be in there and how delicate these samples are to collect. So it's really important that these are held and packaged up quite carefully so we can get them to the lab and they can be identified and dissected while they're in good order. So where are we currently trapping? As you can see, the map shows some of the main locations where we've got traps. We've currently got approximately 38 to 40 traps out at the moment. We're looking to increase that. We're still sending some traps out to locations, and we'd like to thank everybody who's helping us out with those, because as we know, the territory is a large area and we need help to get them out to a lot of areas. So thanks to everyone helping out. And just a point that I want to touch on just quickly is just as we're trapping, it's really important to consider good data management because that then allows us to track where detections are being located, and then this will feed into some further research as we continue monitoring. But just to summarise, the bucket traps that we use are a simple and effective tool. They're easy to use in the field, and yeah, they're a great tool. We use the 2 traps of pheromone lures, and this is to see if we can get any kind of difference in our catch numbers. The trap placement and site selection are critical for our monitoring success. We need to ensure that regular trap clearing and maintenance is conducted. That obviously gives us the best results. It's important to collect good data, and this will be, like I said, important as we keep going. And we're conducting widespread trapping across the Northern Territory with the help of a lot of other people. So thanks very much.
Thanks, Michael. As you can see, they've moved fairly widely across the territory already. if you are seeing armyworm on your property, please reach out. If you'd like to be involved in the surveillance program, please reach out. It's something for all of industry. Biosecurity is a critical part of our business and should be part of your business planning. So it's not something to be scared of, but we do need to get the best available information. So please reach out and be part of the solution. Just want to hand over to Dr. Brian Thistleton now, Principal Entomologist at DPIR. G'day Brian, long time no see.
Thank you, Paul. So I'm going to talk about what we know about management of the fall armyworm, but there will be lots of things that we need to find out in the future. So as I go through this talk, I'll tell you what we can do presently, I'll tell you what we need to know, and at the end I'm going to summarise what we're going to do in a proposed research project on this. So of course, being relatively new, the first thing we need to know are what are the host plants. As Grace said, there are 350 known hosts, but it's mainly a pest of poaceae, the grass crops. In the territory, we recorded it on maize, sweet corn and sorghum to date. We've had one maize crop that was fairly heavily damaged, quite a large crop, which had to be sprayed a couple of times. In Western Australia, I know it has gone on to irrigated rhodes grass, so some of the irrigated fodder grasses. We don't know whether, I mean, it gets on 350 hosts, but a lot of those will be records of feeding and not necessarily a pest status. So we will need to find out what it's going to feed on in the territory and what it's going to be in big enough numbers to be a pest. And of course, we're really keen to find out if it will affect vegetables. We've got a really big vegetable industry, whether we'll get on melons, whether we'll get on cotton. So we have a trial on one of our research days. This is just an initial trial. We actually planted it originally because we were trying to get larvae for our colony. So we planted several rows of sweet corn, which, as you can see in the lower right picture, is infested. And then we decided to check on other crops. We could plant other crops in the plot to see whether the fall armyworm moved to that. And we've got okra, cotton, and watermelon at present. The cotton, we've got both the Bulgard cotton with the BT genes in and conventional cotton. Cotton industry as such is not terribly worried about fall armyworm. They think that the Bacillus thuringiensis toxins from the genes, there are 3 different genes in the current Bulgard, that will control fall armyworm. And in the territory, we've got cotton plots close to a heavily damaged maize crop. In North Queensland, they've got cotton crops close to maize, but we haven't yet found fall armyworm on the cotton itself. So in terms of management, we'll obviously be looking at integrated pest management using a combination of control measures, biological and chemical. We need to monitor populations. We need to make decisions based on thresholds so that we're not just spraying just in case, we're spraying when we get a number of larvae on the crop. Now this is one of our main problems. We do have thresholds for things like maize and other grass crops from overseas, but we haven't developed thresholds for the territory, and that will be one of our research things once we know what the hosts are. And of course, with IPM, you're trying to use soft chemicals wherever possible. So the biocontrol options we've got are 3 different groups of organisms. We've got predators, things that just kill and eat the caterpillars. And we already know from our work on IPM on vegetable crops that the spine predatory shield bug on the left gets to be quite abundant when growers don't spray broad spectrum chemicals, and we've had really good control of Spodoptera lectura and Heliothus or Helicobacter through that bug. So hopefully that will also take out any of the fall armyworm. The minute pirate bug is also another predatory bug. We get general predators like spiders, we get assassin bugs, there's ants, there's earwigs, there's lacewings. There's a whole range of things that build up when we don't spray or we reduce the amount of broad spectrum sprays. Then we have parasitoids, which are also insects, but different to predators. These actually develop inside the insect, so they don't kill it immediately, but they develop through to their adult stage. There's Trichogramma, the egg parasitoid, that's a picture from our vegetable plots at coastal plains. We were going to release Trichogramma. You can buy it from the biocontrol houses, but it did turn up naturally. So Trichogramma is all over the territory, and it does overseas. The Trichogramma pretiosum that we've got here is recorded as a good parasitoid of the fall armyworm. As I say, it's here naturally, but if we had big areas that we needed to increase the populations quickly, it can be purchased from biocontrol agents and released in large numbers. Then we've got a whole load of parasitoids that will attack the caterpillars, the actual caterpillars themselves. What I've shown is aquatic wasp cocoons, probably Coctesia. So those are the cocoons from the larval parasitoid that's bred inside or matured inside the larva. And on the right, there is a fly. Those tachinid flies are also parasites of larvae, and their larvae will develop inside the fall armyworm larvae. And the third biological control option is entomopathogens, so pathogens that control insects. And these are all pathogens that are specific to insects. They're not, when we talk about viruses, they're not viruses that will attack vertebrates or humans, so quite safe to use. You've probably all heard of the nuclear polyhedrosis NPV that attacks Helicoverpa, Virus max. Unfortunately, these NPVs are very host specific, so the one that attacks Helicoverpa will not work on fall armyworm. There is one that does work on fall armyworm called Foligen, which is available in the USA, but because it's a live virus, we can't import it into Australia without doing a lot of host specificity testing. There's also fungi, things like Metarhizium of Ovaria, both of which are used for insect control, and various products are being developed for other insects using these. There's bacteria, the Bacillus thuringiensis in the cotton and the Bacillus thuringiensis we spray in dipel. In that case, we're not actually spraying a live bacterium, but we're spraying the toxins produced. That was all things we need to do in our future research, and at present, where we do get bad damage on our crop, and as I say, it would be mainly maize and sorghum and those sorts of crops, we have to use chemical control. Obviously, being a new pest, there's no registrations for fall armyworm control in Australia, but various industries have applied for minor use permits through the APVMA, and there's a whole load of those. I'll talk about those in a second. But also in the territory, we have the advantage that our Agricultural and Veterinary Chemicals Control of Use Act allows off-label use. So if you needed to use a chemical on a crop, as long as that chemical is registered for another insect on the same crop, you could use it. You can contact our chemical control branch at Berwick Farm. I said there was a whole load of minor use permits. Currently, I did so we brought out a fact sheet a month or two ago, and I think there were 18 or 19 permits. There's been a whole lot more issued since that fact sheet, so I updated that last week. There's now 29 minor use permits, and they're all available on the fact sheet you can see on the right, which lists the chemicals and the products and the crops that are included in the fact sheet, and there's a whole range of different crops. So you can have a look at that, download that from our direct to the APVMA website. The link I've given you there goes straight to their search page for permits. Put in fall armyworm in the search field, and you can see the start of a list at the bottom on the right of the permits that are issued. And if you click on those, you open the full permit and get all the conditions that are involved. The permits are for a range of active ingredients. This is most of them, not all of them, because one or two were there for very specific uses. I haven't included those. But as you see there, they range from different chemical groups. Two things are important there, that the pyrethroids and the carbamates have got very high toxicity to natural enemies. So if we're looking at integrated pest, group, which is what we have to take into consideration when we're thinking about development of resistance. So it's good not to spray chemicals, repeated sprays of chemicals from the same chemical group. So just talking quickly about resistance, it has been recorded that fall armyworm does get resistance to a range of insecticides overseas. We've got labs in Australia that are doing testing for the resistance, and they're actually doing it through DNA techniques. They're looking for the resistant alleles in the DNA to show what the current populations are resistant to. But of course, as we spray, even if we've got populations that aren't resistant to particular chemicals, they can develop resistance as we spray them under selection pressure. So we've got to be mindful of that when we develop our management. And there's also the problem of repeated sprays facilitating development of resistance in non-target pests. So you've got things like Heliothis, Helicoverpa, which does develop resistance. If you've got a resistant management scheme in place for that, and you start spraying things for fall armyworm on your maize, for instance, you're putting extra selection pressure on those Heliothis and compromising your resistance management strategy. So all things that need to be considered moving forward. So just putting that all together, we've got, we're developing a research project to look at these things. We want to look at the host range, as I said, establish thresholds, determine the acceptable stages of vegetables if we find vegetables are a host, and study feeding opposition olfactory behaviour on different vegetables. And we want to look at all the biocontrol options, do a survey of what is locally available, look at what commercially available biocontrol agents are suitable, particularly study the entomopathogens, and I'm very keen for us to look, for example, for the NPV. As I said, there isn't one in Australia. If we can find one here, the company that produced the Foligen in the States also has a branch in Australia, so there may be an interest in the future in using a local NPV, developing that to a commercial product. And also we want to look more at the pheromone traps. We've been using pheromone traps for distribution studies, and we'll still do that. We'll map the seasonal dispersal behaviour and identify factors affecting its movement. So we expect it to move south during the summer, and then during the winter it will be just up through the top end. But we're also going to look and see if we can use pheromone traps as a decision tool to let us know when the moth is there and when we should be timing our controls. And Same details that Praise gave about the contacts. Contact us at DPIR Entomology if you want more information. And the link I've given there will get you into our fact sheets. If you forget the link, just go to our website, search for fall armyworm, and it brings you to the page with the fall armyworm. A whole load of different sub-pages there, but there's a link at the bottom for more information, and that is where all the fact sheets are located.
Thanks, Brian. So hopefully that was informative for everyone. There's a poll there. If you can fill the poll out so we can gather some information around fall armyworm, it would be appreciated. Also, if you've got any queries, please reach out to the department. The department and industry work very hard together in biosecurity, and you know, we've got a pretty strong relationship. So the more information we've got, the better armed we are. So moving on to next week, which will be number 7 in the series, and hopefully I'll be half reasonable at this by the time we get to number 10. But we're going to be talking about workforce. So we're going to have Isla Connolly, our Workforce Development Manager, on next week. We're going to have a couple of guests as well. We're going to be talking about people's workforce demands, what NT farmers have been doing to try and facilitate having enough workers for the upcoming mango season, because we appreciate that's one of the biggest stresses on everyone at the moment. We look like being around 2,000 staff short for the mango season. So we're trying a lot of different ways to address that. We're also going to have a little bit of a conversation about pack houses and social distancing and how we can actually achieve some of those goals in a COVID-19 world without impacting too heavily on your business. So please tune in next week. Thank you very much, everyone. And please remember, we're all in this together. Make sure you ring up your mate next door, say good day over the fence, and make sure they're all right. If anyone's got any problems, please reach out. There are a lot of support services available, so please contact NT Farmers and we'll point you in the right direction of who you need to talk to. So have a great long weekend if you're in the Territory. Make sure you go down to the pub and buy a beer and support a local business. Thank you.